Hey everyone, Gabriel here from Clastify Tutorials. Are you brainstorming for your biology IA and feel stuck? No worries, because today we're taking a closer look into the top 7 biology IA ideas that will provide you with decent concepts alongside detailed explanations. Whether you're fascinated by plants, animals, or other aspects of biology, we've got something for you. So let's start today's journey of discovery right now. And remember, if you enjoy our content, don't forget to like, subscribe, and share your thoughts in the comments section. You will also be able to find a link to an exemplar IA written for each of those ideas. Let's roll! Diving into our first biology IA idea, let's explore the biochemistry behind the rate of protein digestion. Imagine setting an experiment where you investigate how coffee, known for its enzyme-inhibiting properties, influences the breakdown of albumin in eggs. This not only touches on important concepts in food science and biology, but also offers practical insights into enzymatic activities and their implications in everyday life. Picture this: you'll prepare coffee solutions of varying concentrations and observe their effect on egg whites, which are rich in albumin. It's a perfect mix of lab work and analysis, allowing you to see firsthand how substances we consume daily, like coffee, can affect biological processes. Plus, this experiment is relatively quick, making it ideal for those on a tight schedule. You'll measure the digestion rates, provide a clear connection between theory and practice, and even delve into the biochemistry behind enzyme activity. All in all, this experiment isn't just educational, it's directly applicable to understanding more about the foods we eat and our body's reaction to them. And moving to our second biology IA concept, let's focus on plant sciences. Think about this: investigating the germination success of plants, but with a twist, using different types of tea infusions like chamomile and nettle. Each tea, with its unique chemical makeup, offers a natural experiment of how these compounds affect speed germination and early plant growth. It's a perfect blend of botany and ecology, offering a window into how natural herb extract can influence plant life. For those of you passionate about the environment and looking for a project that combines love for nature with scientific inquiry, this is a golden opportunity. Also, if you look into it, it'll be much easier to score for personal engagement. It's also a fantastic way to bring the garden into the lab, or in this case, your home. If you're ready to move forward, head over to clastify.com and it's your go-to resource for finding just the right example to get you started. Check out semantic search for any IB-related content, you'll love it. In our third idea, we focus on analyzing the protein content of different types of milk, including plant-based options like almonds and soy, as well as animal-sourced varieties such as cow and goat milk. This analysis offers an opportunity to apply biochemical techniques to measure and compare protein levels, providing insights into the nutritional profiles of each milk type. Understanding these nutritional components is crucial for informing dietary choices, particularly for those with specific nutritional needs or preferences. This experiment not only contributes to personal health awareness, but also touches on broader topics such as dietary sustainability and environmental impact of food choices. Exploring the effects of sodium bicarbonate of photosynthesis offers a deep dive into plant biology and biochemistry. This experiment examines how different concentrations of sodium bicarbonate, a source of carbon dioxide, influence the rate of photosynthesis in aquatic plants. It's a practical investigation that combines theoretical knowledge with experimental observation, highlighting the process plants use to convert light energy into chemical energy. By analyzing the impact of varying CO2 levels, students can uncover the conditions that maximize photosynthesis, linking the environmental factors with biological outcomes. This experiment stands out for its straightforward approach to complex biological processes, making it a compelling study for understanding the interplay between plants and their environment. Moving to our fifth idea, the role of hydrogen peroxide in plant germination, the simple molecule H2O2 is a bit of double-edged sword for plants. It's not just a potential threat as a source of oxidative stress, but also a beacon for growth, signaling the beginning of life from a seed. By exploring how different concentrations of hydrogen peroxide affect the germination rates, we're tapping into the core of plants' stress physiology. This isn't just about seed sprouting, it's a deep dive into how plants confront and adapt to the challenges posed by their environment. What makes this study so compelling is its broader implication for understanding plant resilience. Through the lens of hydrogen peroxide's impact on germination, we're uncovering the intricate ways plants navigate stress and adaptation. This research isn't merely academic, it's a glimpse into survival strategies of plant kingdom, revealing the delicate balance between growth and survival. In essence, by decoding the effects of hydrogen peroxide on germination, we're learning more about the resilience and adaptability of life itself. In our sixth idea, we delve into the microscopic world of yeast to understand how sugar solutions affect its respiration. This project is a treasure trove for those fascinated by the unseen forces of biology, offering insights into anaerobic respiration, a vital life process. By experimenting with various sugars, we're not just observing yeast at work, we're uncovering the secrets of its metabolism and its pivotal role in cellular respiration. The beauty of this study lies in its practical implications. It bridges the gap between biology and industry, from baking bread to brewing beer, demonstrating how understanding the fundamentals of yeast respiration can have far-reaching applications. This investigation not only enriches our knowledge of cellular processes, but also highlights the incredible impact of microorganisms in our daily lives and industries. Our final exploration brings us to the dynamic world of osmosis through a hands-on simulation. By using simple potato strips immersed in different sodium chloride solutions, we get to observe osmosis in action, witnessing its effects on cell mass firsthand. This experiment is an excellent way to visually grasp the principles of osmosis, bringing to life a fundamental concept in cell biology that's critical for understanding how cells interact with their environment. I encourage you to visit Clastify for a detailed example of this experiment. It's not just about watching potato strips change in mass, it's a deep dive into the osmotic process that sustains life at the cellular level, offering a clear, tangible understanding of how osmosis works and why it's so important in biology. And that concludes our exploration of top 7 biology IA ideas. Each of these ideas offers a unique avenue to engage with biology, from cellular processes to ecological interactions. Remember, these are just starting points. Your creativity and research will bring them to life. For more guidance on IA exemplars, check out Clastify's website. If you found this helpful, don't forget to subscribe and share your thoughts below. Until next time, keep exploring the wonders of biology.